stem cells upregulate a cytokeratin, migrate, divide, and differentiate in the small airways. And they form these kind of epithelial scabs that restore barrier function, but really at the cost of lung function. And in this dysplastic epithelium or bronchialized regions, what you see are three major cell types: goblet cells, club cells, and those famous Tuft cells we've heard so much about today. And as it was mentioned earlier, they're so-called Tuft cells because of these apical projections that they use to kind of sense the environment and trigger different signals. Much of what we know about Tuft cells actually comes from work in the gut, where Tuft cells regulate this ILC2 circuit in response to parasitic pathogens. Interestingly enough, Tuft cells are very similar to taste buds, as we've learned earlier, and they express many similar receptors, one of them being TAS2R, which senses bitter substances, quorum-sensing molecules from Pseudomonas, as well as apple cider vinegar, a very popular drink around this time. The other receptor that Tuft cells express that's unique to them, unlike taste buds, is a succinate receptor. And they use this to sense the metabolites of parasites. And once activated, Tuft cells release IL-25, inducing ILC2 cells to release IL-13, acting on stem cells to differentiate into Tuft and goblet cells, and a positive feedback loop. And this all promotes that weep and sweep phenomenon that we discussed earlier. Because as it's been shown at earlier talks today, Tuft cells are actually found throughout the body, especially in the upper airway where they sense air allergens, molds, and viruses, as well as the lower airway, where they sense viruses upon severe injury, and finally the gut, where they sense, along with parasitic worms, bacteria, and protozoa. And in response to these various inputs, they have specific type 2 mediators, the IL-5 cytokine, which is its primary output, as well as these lipid mediators, leukotriene C4 and prostaglandin D2. As was mentioned earlier, Tuft cells also secrete acetylcholine, which is involved in neurogenic inflammation as well as mucociliary clearance. And believe it or not, that cough, or perhaps that was a sneeze we just heard. So in the upper airway, it's been shown that they regulate this mucociliary clearance, increasing B frequency to clear pathogens, as well as amplifying allergic response. And in the gut, it does the corollary to that, a weep and sweep phenomenon to expel these parasitic worms. But the role of Tuft cells in the lower airway is largely unknown. And before someone asks me, no, the Tuft cells in the lower airways are not innervated. Okay. So the model that we use in the Vaughan lab is a flu model of ARDS. So this is not your man flu, right? This is a severe form of the flu that results in massive lung inflammation, severe hypoxemia, and most importantly, similar to human ARDS, there's an acute and prolonged period of alveolar capillary disruption. And to do this, we intranasally infect mice with a mouse-adopted strain of influenza A called PR8. And you can see over here, the x-axis, the y-axis is body weight, and x-axis is post-day's infection. And you can see after infecting these mice, they have nearly a 20% weight loss by around day 10, and then they do slowly recover by day 15. And really, this is a proxy for lung injury and severity, as well as inflammation. You can also perform pulse oximetry on these mice, and you can see that there's an oxyhemoglobinator around day 10, down to around 65%. And then finally, if you dare, you can perform bronchioalveolar lavages on these mice. And you can find that there's an increase in protein, indicating a decrease in this alveolar capillary barrier. That's both at day 10 during that acute phase of infection and later on in day 21 during that recovery phase. So if you take these mice after infecting them, monitor their vitals over time, and harvest their lungs and stain them, what you find is something like this. So those are these dysplastic keratin 5 regions that are those scabs I was talking about. And these rarely become appropriate alveolar cell types and rarely engage in gas exchange. Labeled in red are surfactant or alveolar type 2 cells using surfactant protein C, which proliferate and generate gas exchanging regions. So really what you're looking at is areas of injury that's never really truly going to repair or recover, and areas that have recovered and repaired. And if you zoom in on these regions, what you find are Tuft cells in these dysplastic regions. And you can see here, they're lined up like good soldiers, looking into the airway and sensing what may be coming by. But if you look more deeply, you can see they're also swimming in these keratin 5 regions. And you can see they have their classic morphology with this perinuclear clearing. And of course, the question is, why are they here? Why are they in the lower airways? And what are they doing? So the first thing we wanted to do was prove that these are bona fide Tuft cells. So we used bulk RNA-seq using that famous trypin-5 GFP mouse line. And what we found is that these Tuft cells have a transcriptional profile that's very similar to intestinal Tuft cells as well as tracheal Tuft cells, and that they express major Tuft cell markers, including SPB, SOX9, trypin-5, and DCLK1. So we didn't just show that there are Tuft cells down there. We showed these are Tuft cells that are very similar to the rest of the body. And with this in mind, knowing that their transcriptional profile is similar, we hypothesized that perhaps these cells are regulating an ILC2 Th2 circuit in the lower airways that's similar to what occurs in the upper airways and in the gut. And so just to drill into this a little bit, because this is kind of the basis of this story, is that in the upper airways, similar to the gut, it's known that Tuft cells sense different viruses, including rhinovirus, and upregulate the secretion of IL-13 and 5, which can just drive this allergic disease state marked by eosinophilia, mucus metaplasia, and airway hyperresponsiveness. And so we thought, if we infect mice lacking Tuft cells, this pathway would be abolished and they would have an attenuated response. But unfortunately, like so many things in science, that's not at all what happened. So you can see here is the y-axis is weight, and x-axis is days post-infection. The black circles are our het controls, and the open circles are our knockout mice. And you can see by day 9, they've lost almost 30% of their weight, similar to the other group. And then there's this recovery phase that occurs around day 15. So we're really surprised by this result. But then we thought about it a little bit more, and we realized that the ectopic Tuft cells really don't form until day 15 or 17. And so it makes sense that they don't play a role in the acute infection because they're not even present. So this raised 3 intriguing questions. What is the role of Tuft cells in amplifying type 2 inflammation following ARDS? Is there a role for Tuft cells in development of asthma later in life? And then finally, is there a role for targeting type 2 inflammation in influenza-induced ARDS? And for the sake of time, I'm just going to focus on the first part. So like all good scientists, the first thing we did is we took a circuit and we broke it to try to understand how it works. And what we did is we systematically disrupted these key players in this pathway to see if there is a functional role in the lower respiratory tract, starting with the brakes. That's interferon gamma. And so this is an immunofluorescent staining of a control mouse and a mouse, interferon gamma knockout mouse, day 22 post-infection. And what you can see is there's a statistically significant increase in the number of Tuft cells, which is what you would expect to find if the circuit is functional in the lower airways. Additionally, we also found an increase in IL-5 at day 9, as well as ILC2s at day 9. So we thought this is great. We can check a box there that interferon gamma seems to be a part of this circuit. Next, we moved on to focusing on the role of ILC2s. And so before I go into this data, I just want to thank Jorge's lab here on campus who gifted us these mice. They're really unique. They're not truly an ILC2 knockout mice, rather they're an ILC2 deficient mouse. They lack this ID2 transcription factor, which results in reduced development of ILC2 cells. Nonetheless, it's treated as a knockout model. And what you can see here is that in ILC2 deficient or knockout mice, there's a decrease in the number of Tuft cells, both per mouse and then per histological slide. There's even more of a statistical significant decrease. And just to show you, we did do some flow cytometry and showed that ILC2s are decreased in the lungs of these infected mice. So finally, we turned our attention to the receptor of this pathway, Tuft cells, where we hypothesized that without Tuft cells, you would have a decrease in some of the readouts of these pathways. So we infected mice with and without the flu, performed a single cell suspension from their lungs, did immunostaining and flow cytometry. And what we found was a statistically significant decrease in the amount of eosinophils. So you can see here, there's about 1.5 to 3% as a percentage of CD45 cells. And in terms of absolute numbers, it's a little under 250,000 compared to a little over 500,000. We also found that there's a decrease in ILC2 cells, which is exactly what you would expect because this is that positive feedback loop. So kind of stepping back and putting all these pieces together of this puzzle, what I've shown you that is if you delete interferon gamma, you have an increase in Tuft cells. If you delete ILC2 cells, you have a decrease in Tuft cells. And then finally, if you delete this receptor, you have both a decrease in ILC2s and a decrease in eosinophils. And so there's just one component that's missing from this story, and it's that these key cytokine mediators. So if we were to prove that this ILC2-TH circuit is functioning in the lower airways as it does in the gut and the upper airways, these cytokines would be the key mediators. So the first thing we did is we infected mice that lack IL-25, and what we found was no difference in the number of Tuft cells in these dysplastic regions. We then infected mice that are IL-4R alpha knockout, which is used by both IL-13 and IL-4 cytokines, and again found no statistical significance difference in the amount of Tuft cells in these regions. So going back to that ILC2 circuit in the gut, what I think our work is showing, contributing to this growing literature, is that Tuft cells operate at an IL-25, IL-13 axis in response to certain viruses. And we believe that's what's happening in the lower airways. So we then turned our attention to an unbiased approach using single-cell sequencing to see what pathways Tuft cells might regulate in addition to an ILC2 circuit. So we infected mice both with and without Tuft cells. We performed cell sorting and bin them as either immune cells or non-immune cells and perform single-cell sequencing. And so this is a UMAP of the result of just the non-immune cell types. And what you can see in aqua blue are Tuft cell deficient mice, and in orange are Tuft cell sufficient mice. And overall, you see right off the bat that there's a lot of overlap. But there are unique gene expression profiles, especially in the alveolar fibroblast and the GCAPs. But if you really begin to look more deeply at these cells and bin them into epithelial, mesenchymal, and endothelial cells, you'll see a pattern where the top differentially expressed genes in Tuft cell deficient mice show evidence of Th1 pathway activation. That is, epithelial cells upregulate the expression of retinal LA, which suppresses Th2 immunity. Mesenchymal cells have an increased transcriptome expression for CXCL15, which is a neutrophil chemokine, and concomitantly, endothelial cells have an increase in CYP1A1, which is a gene involved in combating oxidative stress, which is exactly what you'd expect if there was more neutrophils. If you perform a gene ontology enrichment experiment on Tuft cell deficient mice post-flu, you see three major biological contexts are upregulated significantly: response to oxidative stress, cell proliferation, and Th1 immune response, that's CXCL15 and retinal A. And in fact, we actually have in vivo data that supports this in vitro results, where we infected Tuft cell deficient mice and then challenged them with Alternaria. So this is a classic allergen model. And what we found, surprisingly, is that in these mice that lack Tuft cells, there's actually an increase in neutrophils. And so what this suggests to us is that ectopic Tuft cells may balance Th1 and Th2 immunity. That is, Th1 immunity is marked by neutrophilia and tissue destruction versus Th2 immunity marked by eosinophilia and tissue repair. So the three major conclusions from my talk is that Tuft cell microbial or viral interactions are well characterized in the large intestine and the upper airways, especially in the context of Th2 immunity, but we've shown this may be a Th2 independent pathway in the lower airways. And it's incompletely understood. And then finally, the presence of ectopic Tuft cells may promote a Th2-mediated tissue repair upon subsequent injury. And in fact, there's a great deal of literature that suggests that there's an increase in IL-5 secretion from ILC2s, recruiting eosinophils during the recovery phase following an influenza infection. And with that, I'd like to thank the Vaughan Lab, especially Alona, who is with me today, and it is her birthday, very special day. T32 Leadership, Lisa Young and Sharon McGrath, my SOC committee, the various funding sources listed below. And with that, I'll take any questions.
So I don't know if this piece is a little bit downstream, but at first you said that the scar, the repair, involved 3 different types of cells.
Yeah.
And I wonder if there was any communication from the Tuft cells to the goblet and the club cells. And I don't think that's not your focus, but I wonder if you can speculate.
Sure.
That.
Sure. Certainly, kind of not shown here is because we showed that IL-13 and IL-25 are not the key Th2 cytokines, we actually created air liquid interfaces and treated them with various cytokines. And we do see that there's an increase in goblet cells and those ALIs in conjunction with an increase in Tuft cells. So I think there certainly is a relationship between them. I don't know if it just is, they both respond to similar stimuli in similar fashion, but at least in the upper airways, the role of goblet hyperplasia and Tuft cell activation is well established.
Anybody else? No.
Yeah. So what do you think would happen if you did repetitive viral infections? You let them recover, then you hit them again with the virus. And you're going to see more and more Tuft cells building up in the lungs. And I guess the thing that I'm not following is, why don't you think this is a driver for the production of asthma? You've got these resident Th2 because they're not secreting the IL-13. To me, it makes like this is like a perfect segue between repeated respiratory infections and the development of asthma.
Right. It does fit into that paradigm of the Tucson study, which shows viral infections early in life portends the development of asthma. And Mark Hirschison and others at U of M have actually performed those experiments using rhinovirus and young mice. The reason why we don't think this is actually a true Th2 allergic asthma model is because they didn't show you there's no signs of eosinophilia or an allergic phenotype. What we do see is this dysregulation in the absence of Tuft cells towards neutrophilia. I think what we're seeing when we do see eosinophils there, they're actually participating in tissue repair and recovery rather than that historical context that you have eosinophils, therefore you have this robust asthma response. I think in the mice that get repeated flu injuries, similar to mice that are vaccinated with the flu and develop breakthrough infections, that you would see in mice that are Tuft cell deficient worse outcomes. And so I think that part of why we don't see a difference is the way we kind of set up the experiment. And I think that these Tuft cells help with recovery and repair. And the best way to test that would be to do these repeated flu infections or do flu, vaccinate them, and so forth, and to get a really robust breakthrough infection as others have done. All right, thank you so much.